hoping to show you what to do is how to make your own Petri dishes really simply and easily. And I'm going to show you also then how to take a small piece of a mushroom, because a mushroom is actually the mycelium or the hyphea, the small microscopic pieces that make up the mycelium, is also the mushroom. So you can take a piece of the mushroom, and if you can get it to start growing, the tissue to start growing, it's basically 100% mushroom. That probably doesn't make much sense, but
it does.
So that's what I'm hoping to achieve today. So it's a really simple hydrogen peroxide agar. Basically we have 200 mils of water. We have 1 teaspoon of agar. I probably should have left my glasses on. So 1 teaspoon of agar. Oh, that wasn't very professional. And then we want 1/4 teaspoon of molasses or honey. And we also get a pinch or just a very small amount of yeast, just a pinch. Basically like you would salt, I suppose. You can use Vegemite if you don't have yeast. And just start mixing that. Oh, we bring it to the boil. Not sure how that's going to go in the wind, but we'll give it a go. You could Google that yourself.
All nutrients, so basically everything that the fungus requires to feed on is in that prep, in the medium. So a bit of sugar with the molasses, the yeast has got all like vitamins and all kinds of stuff in it.
One of the things they said to not do is to not boil it too much, to just bring it to the boil, as by boiling it too much you may change the sugar that you're trying to get them to live. You might caramelize it, I guess, or change it. It won't be so easy for them to eat. So a bit of a waiting process, watching a kettle boil. You could probably make it quicker if you'd boiled your water first, but it's not going to make any difference to your sterilisation.
Is that the aim, to sterilize, to make it sterile?
Yeah. So you're bringing your water up and boiling and
kill everything else.
Yes, and then what you do is you wait for it to cool to 50 degrees and you add 2 mils of hydrogen peroxide, and you then instantly pour it into your dishes, your clean dishes, and that's it. Very simple.
So cool.
Yeah. Very simple. Yeah, I did have a spoon, I thought there. All of this stuff I have wiped down with a mixture of 70% metho and 30% distilled water to sterilise it. There we go. Just get in there. I try to watch it a fair bit so it doesn't boil too much. And then this, here it goes.
Starting to.
So even my cotton cloth that I have here is soaked in the sterile solution just to maybe eliminate. I shouldn't talk over the top of it either. I wrote that note to myself.
Wear a mask.
Yeah. I'll wait for it to boil. Okay, it's boiling. To me, that's boiling.
Yep, that's boiling shitloads.
Contaminate the water.
Yeah, or we were just talking before, Gilly and I love doing stuff together, and every time we do Petri dishes, we laugh and talk so much they're just contaminated. There's that. And you then get this, and we did this earlier, and it did take a little while for it to cool. So we just pop that in there and wait for it to drop back to 50. It's going to beep at me for a minute. So while we wait for that, we made some earlier. Here they are, already set. And less than half an hour ago, I think we poured them. So
when you say Petri dishes, is that what you mean? It's a little plastic container.
you can use whatever. If you could afford glass Petri dishes that you could sterilise yourself over and over.
Yeah.
But we're doing them to send off to Roger to be DNA'd.
Yep. So you don't mean you're making a Petri dish out of glass?
No, but you could
Yeah.
keep
It's all right.
recycling
Yep.
them. Yeah.
Yeah.
To me, this is a Petri dish.
Yep. No, very
Yeah.
good. Excellent. You're making a culture.
Yes.
Yes.
Yep.
I'm making culture. So here we have one sterile ready to go. It's cooled down. This. Now we have, Gilly has a little mushroom, very tiny. I think it's going to be a bit annoying. We have this, a knife, and tweezers. So, sit down here. We will first of all sterilise the knife.
Whoa. It needs a Bunsen burner. That's cool. Is it like a blowtorch? What's it called?
Baking. You know, the women candy the.
Oh.
Yeah.
Wow.
Also do the same with your tweezers if you're using them. I find it's easy to have something to pick it up with, rather than otherwise you end up using your fingers. I have also used hand sanitizer on my hands today to maybe eliminate my own human glasses. So this is a little bit perished, but we'll give it a go. Basically, they suggest you slice the mushroom in half. It's fairly perished. Okay, we don't actually need much, but this one's fairly old, but we'll give it a go anyway. And the thing with Roger is, because he's a macrofungus guy, he's not going to care what fungus grows.
Microfungi.
Yeah, if
microfungi.
a microfungi, because then he might discover some new microfungi from your rotten mushroom.
Yes.
So he will be happy anyway. This isn't the best cutting dish. You literally only need... I've got a feeling it's a bit too decayed, Gil.
Do you want my ones that are over?
No, it's all right. We'll do it anyway. So you literally only need the tiniest little piece. They suggest to try and get a small triangular piece, but that's not always going to happen. And you just place it gently on top. Don't push it down. Seal it up. Get your Nico. I think we're up to #6. Give yourself whatever number you're up to. That's it. That's sterile. Don't reopen it and wait, and you will find in a few days there will be some kind of activity happening, like in these ones. My camera. So pass them around. I've made up, as Roger emailed me because of the ones I handed in last time, he gave me a list of what he likes to know. So I've printed up some forms for people as well. So I've just gone What number have we got? Hashtag. So this is hashtag 1 sample. I've done hashtag 1. Substrate, source, host, the collector, the date, the location, and any additional information you'd like to add. And I'm hoping when I send that to Roger, he can see that's hashtag 1 on the on my Petri dish, not a glass Petri dish. And hopefully that's the information that is enough. Oh, that's all he asked for last time. So yeah, I've started. We're up to, that's hashtag 6. So we'll have to write here, open it up. We'll go, it come from your place, didn't it, Gil?
Yeah.
So hashtag 6. It was swamp mahogany, isn't it?
Swamp mahogany,
Swamp,
gymnoparis.
yeah, swamp mahogany. Possible, they're called gymnopilus, that mushroom. They're an orange mushroom. They are they live off dead wood, saprotrophic or whatever that word is. And they also have, they're a magic mushroom, a native magic mushroom. Possible gymnopilus.
I find them on the swamp mahoganies at home.
So Gillian
And
Smith
the
is a
koalas
collector.
leave their babies at my place in this swamp mahogany grove, and to koalas, swamp mahogany is like it's like chocolate to them, the leaves of them. So I'm starting to want to know whether this being psychedelic, is it in the tree, is it in the leaves? Are the koalas leaving their babies there to be off their heads while they do the separation, the weaning? You know,
Very
calm
possible.
them down and yeah.
Amazing.
Do you know what the like order is of the swamp mahogany? Like that's not a eucalyptus or a gum tree.
I
what
could
is
probably
it?
tell you.
Yeah,
off the-
Gillian's
but I
pretty
have
good
to
at stuff
go
like that.
look
Anyway,
at
let's
it,
just
yeah.
have a look at if he's wanted to see.
Track it down. Here we go on our temperature.
So that's basically, the temperature. It did take a while before.
Yeah.
Enough times it kept turning itself off. So that, guys, is basically it. And if you wanted to do what we did with Roger, where you go to a tree and get the sap or inside the fruit, these little dishes are going to do exactly the same thing. And you can post that. You could do your favourite lily-pilly tree like we did with the fig tree or whatever. But it's a great time of year. There's plenty of fungus around. Good time to build Roger's collection up.
Mm-hmm.
So that's it. Except when this hits 50 degrees and it's at 70, so we're still a probably 5 minutes off. When it hits 50, I will get this and
Oh, yep. So hydrogen
syringe.
peroxide.
Here.
Ah, there's one here.
oh, thank you.
Sorry, I'm not sterile.
Okay.
Yeah.
Yeah, right.
The hydrogen peroxide is very sterile.
Mm.
And possibly what would be better is if you put that in a separate container, but I'm just going to go with this. 1 to 2 mils, it says, as if you were doing it for cultivation purposes, you would learn that the pink oyster doesn't particularly like a big dose of hydrogen peroxide, so you would probably only add 1 mil, but it doesn't really matter, we just want it sterile, so I go 2 mil.
Just in case.
Just in case. Yeah, so when that hits 50, we shall add that.
Now, would it be, does it help at all if there's less settlement by covering maybe with a sterile dish like on top of that while it's
They didn't do
cooling?
that.
No.
I think it's about your wind factor.
Plus you end up putting the hydrogen peroxide anyway at the
Yeah,
end.
that's right, at that
Yeah.
50 degrees, and then you're pouring it straight away.
Exactly, so
it's
really,
basically getting another hit of sterilisation after being
Yeah.
boiled, and then you're pouring it.
Yeah.
So how do you get the samples to Roger? You just mail them?
Yes, he gave us a mailing address,
Yep.
and yeah, we just send that off.
Do you need any special packaging or insulation or?
Well, I'm figuring these egg cartons, that's the other reason I really love this size.
Ah, right, yeah.
That's
That's
because
cute.
I did buy another size, which I was going to trial today, but I just think this is going to be perfect.
Okay.
Just post the egg carton.
That's a great idea.
Yeah.
So, he's happy for all of us to send stuff in?
Yeah.
Okay.
He
Great.
wants to
they have a
develop.
blog. They have a thing actually on his webpage that says, "Please send us stuff."
Okay.
Yeah.
So you
Because
share
there's no
those details?
macrofungi, so like mushrooms and brackets, they don't have a collection of that.
Yep.
So they're only doing microfungi like we did with the leaf samples.
Yeah.
So this is like going to increase our knowledge about fungi,
Talk about
which we
this
know
too.
5% of in Australia at the moment.
Can you share
5%.
those contact details on them?
Yeah. I can probably send it via our newsletter.
And like if you pay for things to be DNA, the cheapest you can get it for in Australia at the moment is $20 per sample. Roger is going to give us the DNA results for nothing. So if it costs you $20 to get a lot of them posted there, you're still in front.
And we're really contributing to that,
Yeah.
knowledge of mycology that is just
68.
non-existent in our country. And like yet, like we rely on our very breath depends on fungi pretty much, like it's crazy to not know about them.
So are we collecting fungi pretty randomly and then send them off?
If you want.
Yeah. But one of the
As long as
Or
you
do
have
I
that.
mean, you know a lot of fungi already, so you probably try to avoid the ones you know.
You can, but Roger has very few guild
Yes.
fungi.
or
And he
polypores.
wants,
Yeah.
wants more mushroom fungi and visible, because he's got a huge collection of the microfungi,
Yeah.
but he wants more Australian fungi. And you can go, if we found an edible merismus or something really special, like a woodier, that's so expensive now to buy, and it got DNA'd, we can ask, as long as we don't make money, we can ask for that tissue culture to come back to us, and then we can go through that process, like I talked about last time, where you get your Petri dish, get it going, put it in your sterile grain, get that going, then grain and straw, and then you're eating it.
Plus, so by doing this, you're giving a live sample as well, which it doesn't happen right now. So if shit hits the fan, which it will, we now are putting this, and this is stored for generations to come, a seed bank of fungi that are going to help when climate change is coming, like, when there's, I suppose, recovery, post-recovery, new worlds, like new world, like recovery.
It's been happening since 1910, but don't worry.
Yes.
the
So
world's
basically, it's
still
about
here.
also having these cultures alive so that they're actually there, because we're losing them. We're losing everything, basically. So this is a way of doing it. And I'm not sure how good it is with cryo fixing them.
We'll probably be gone too.
Sorry?
We'll probably be gone
Yeah,
Yeah.
I'd say so.
But let's say some of us survive. There's that availability of that.
And
I
we
know
have the website.
if the websites do, even if they do. Look, we're just trying our best to contribute something as a citizen science
Yeah.
project. So this is, I think, like a very worthwhile endeavour.
And he's really happy to take them. Like when I asked last week, he's really happy. And then he sent me an email saying, please send whatever. The other thing I learnt in the course is when you see Ganodermas, the hard bracket fungi growing on trees, and people usually remove them to get them identified or to send to the herbarium, and might only be one there.
And they might be hundreds of years old because they grow really
So you can
slowly.
sterilise toothpicks. I've done bamboo skewers. I did them in the pressure cooker, and then I've left them in an alcohol mixture to keep them sterile. You pierce near where it's attached, leave it there for a couple of weeks, come back, pull it out, chop off where the mycelium has started eating it into your Petri dish, and you have a tissue culture without removing your Ganoderma.
Beautiful.
Which I think is one of the amazing things that I learned out of
Yeah.
the course.
Would that
Is
work with a lot of the other?
What about those big hoof things? The ones we
Yeah.
wanted to get for the hats and stuff?
Yeah, the
Even your
felinus,
normal
yeah.
separate wood-eating ones.
Yep.
Saprotrophic, yeah.
Yeah.
I don't know if they're there long enough,
Yeah.
if you know what I mean.
Yep. Yeah.
Because they rot down so much quicker, don't they?
Yeah.
But maybe with the panis and stuff like that.
The hardier ones.
The
yeah.
hardier ones.
Yeah.
Yeah.
And I also feel like this technique allows us to not even remove mushrooms at all. What we could do is go around in nature and take, because we only need a tiny sample for it to grow. So we don't actually have to take anything.
No, you can just split it. Say you're out in the field.
Yeah, and it's still got the spores, producing that
Yeah.
for the environment. Because sometimes there's only one mushroom you see, and we just take it and collect it normally to put into the herbarium. But we're destroying something that might come once every 10 years, and there's only one of. Like, there's really no consideration sometimes for scientific endeavours to actually look after nature and actually not just take from it. So I feel passionate about this way of, like, let's clone it, because essentially this is a clone now. Like, all these are clones
One at a time, so we
of
don't
that
hurt him.
particular species, you know, of that particular
sitting there then.
individual. So, yeah.
I'm just trying to remember where I saw a woodier last, but I think it might have been in
We can
Mycology
buy it, but it's gone
Park.
up to $74.
No,
wasn't it?
I mean in the
was
forest,
expensive.
but I think
Oh,
it
yeah.
was Myala National Park. Like
We
it was
last
everything.
wood year we saw Maroochydore River was the most amazing spot. We'd never seen ferns like it on the salt water. And there's a mangrove fall and collapsed, and the whole thing was wood ear.
It's lovely.
We should have stolen that.
Taking the whole tree.
But a
home.
lot of this, if we get it, like we were
Some
just
private
talking
land.
about on the way down, because we've been into the mushrooms for like 4+ years, going exploring from. But it's only really the last couple of months I've learnt this bit. And now I'm like looking at all the things that I'm learning are edible that we've seen. And like I've been back there recently because I have a motorbike now, and do you think I can find them? No.
No, because they're so ephemeral.
But
Yeah.
everything has its cycle.
Yeah, yeah.
No,
yeah.
everything has its cycle
Yeah.
so quickly. It's really interesting. It's like that oyster tree we went back for 1 year later.
Yeah.
We were lucky to find it. It just dissolved.
Because that's what they do. They eat the wood and then they disappear. The spores go off somewhere else to look for a different piece of wood. You know, that's their
Well,
role.
we don't know. Some of them could be living in it from its seed. You know how that's
They're
part
all
of
the
it.
time
They're,
just
they're
waiting
all
for
the
the-
Yeah, the
time
spores,
waiting
yeah.
for that tree to die.
Yeah,
It's just part of their. We're at 60 degrees. So we have 10 to go. Sorry, guys.
that's okay. That's good.
So, what I was hoping, if you felt like doing, you could go look for a mushroom. It doesn't have to be a mushroom. You could get the sap like we did that other time as well. And I've got some distilled water, and you could, you know, try and grow microfungi for Roger. As long as you write down on that bit of paper your number, where it come from, and that's it, really.
Mycelium would be interesting to find on leaves and stuff, where it's already
that would be
propagating the
cool.
leaves, you know, like that would be really cool. But also I've got a few that I brought, like two species that I brought in that we could try as well. And we could either do the rainforest around here and go for a walk, you know, walk and have a look, or go to the butterfly walk where there's much more diversity. So
Yep.
up to us how much time we want to spend on this.
Well, you guys can do whatever you want with them.
Cool.
Yeah. And this is the other thing. I haven't practiced this yet, but we learnt this in the course. You basically get a small piece of mushroom, same as that, very tiny, and you put it in this thing, which is called an Eppendorf tube, but mine's probably a cheap version. And you get silica beads that change colour when they've gathered moisture. And you fill this with your little tiny piece of mushroom, and when you see the silica beads have gathered the moisture, you empty it out in a sterile environment, and then put your mushroom that's become dry from the silica beads taking all the moisture out, clip it up, and post that off for DNA sampling. So I haven't practiced that a lot, probably mostly because I'd have to pay $20 for each one, or send it down to the Haddon Community Learning Centre, which might not hear back from them.
So the color change allows us to see that the moisture has been taken out
Yep.
of that little atmosphere,
Yep.
and
you-
therefore the mushroom is dry. So
Yep,
this is
and if
why.
you think that the mushroom hasn't dried, you add more silica beads and leave it another couple of days and check again
Perfect.
until basically your silicas aren't going to change colour. They've taken all the moisture out.
And the reason why we do that is because, you know, molds will eat the decaying mushroom if it's moist. So we want to preserve it from not being decomposed by molds and other fungi. Because
And to
of
do the
course fungi
DNA,
get
they
eaten by
grind
fungi.
it up. They
Yeah.
grind it up.
So it's
and
got to be
then-
just that. Otherwise, you've got many, many different forms of DNA from different species. So you want just the one species that you plucked, and that's how you know that's what you're getting. So yeah,
When you say mold, that's another fungus.
it is.
Yeah.
But if you want to, you know, get that one identified and you're paying for it,
Yeah.
you don't want your contaminants to also be there. Although contaminants are often what science is about, like penicillin. It's all about contamination. That it's an accident, or you know, you end up finding quite an interesting discoveries as a result, making discoveries.
57.
It's all right. 7 to go. Should we do that one? The little, what was it called again? Polyporous.
Does anyone remember that?
I've done that, you know. I'll show you.
You've got to record.
It's
every
here.
time
Hashtag 4.
it's been raised
Where's
a
mine?
degree, and you've got to measure the time it takes to raise the water 1 degree, and then plot it on the graph.
Wow.
That was exciting.
I think I've got it going.
Literally watching.
I think it's going really well. See, can you see the white?
Yes.
It's exactly the same species. I had it on a eucalyptus branch at home.
I don't think I went as far in physics.
I wonder if there's a difference between them, like the DNA, whether there's
Yeah, well, we could do yours too.
just to see whether what happens, you know. But yeah, it might be a waste of money, but I don't know.
Because they're quite hard to do. That's quite dry too, isn't it? It's really dry. That was
Yeah.
fresh, that one.
Yep.
These
Should
might
we-
be a little bit too dry.
Okay.
You know, if you know what I mean?
Yeah,
Like the
sure.
myceliums died.
Yeah, yeah.
Yeah.
They were still alive last night. I should have put them in water. I've got some in water as well.
Okay.
But also we've got the stinkhorn. We could just try it. Maybe do a
Bloody
spore
hell.
dilution. I mean, it would be amazing if
It
we
would
got
be
into-
amazing. And it stinks like dog's dick.
Yeah. I think dog's dick doesn't smell that bad.
Oh, I think it
It
does.
smells like shit.
I don't know, but these things are gross.
Oh yeah, we've got those in the community garden.
Okay.
They cried up.
I'll do it. I'm happy to play with shit.
I just find it interesting, but we can only have a go.
We can do it. What are you trying
Okay.
to do with my...
I'm gonna film
recording. I'm recording.
Yeah, I'm not going to film you do it.
Oh, okay.
You
You
can't-
don't want to do it.
Well, I'm not sterile.
We'll just use the hand
Have you got
sanitizer.
new ones?
Yeah, they're all brand new.
Okay.
So that would be #7.
All right. So, where's your little Bunsen?
Yep.
All right. You tell me how to do it. Yep. And then you go.
Yeah, don't
this.
burn yourself.
Do you wait till it goes really hot, like as in red?
Yeah. Well, I mean, it doesn't have to get that hot, really, does it?
All right. Let's turn it off.
That's enough. What about your tweezers?
Yep. I'll do that later when
Okay. I'm just gonna
once I've cut. Do one thing at a time.
Okay.
Just wait till it cools down a bit. This is my little Petri dish.
You could wipe it on the alcohol wipe, on the microfiber cloth. It might be wet enough to.
Oh, just wait. All right, ready for the stink. And I think we should do a spore one as well.
I didn't really notice that they were that stinky at all, really.
Oh, they are.
Did you
Wrong
get-
way round, darling.
Is this the...
That's it.
Yeah. That's cool, because now I can just cool it down on this. Doesn't smell
It doesn't
right now.
smell bad yet.
Actually, tweezers would be good, because I could rip it apart, to be honest. It's not good with this thing, hey?
It's a really soft product.
If I can do both... I call it the alien butthole fungus.
Where did you find that?
My friend's growing it in his backyard. Ooh, I got a nice...
Yes, it's a
one.
very common one.
This is nice.
The Asians eat them not like that. They have they start as an egg, like a perfect little egg,
Yes.
and they call them dragon eggs in China, and they're very highly sought after. They're highly medicinal, and they eat them while they're like that before they erupt out. You can Google that. They have recipes and everything on there for them. I haven't got that brave. And you'd be surprised how tiny the little sample can be.
I wonder if that's how a lot of species go extinct. When humans keep eating the spores of these
No, I'm going to do a spore one as well.
But it should come, if we didn't have water treatment, waste treatment plants, it would be coming out in the human shit and would be still doing the cycle.
Oh yeah, yeah, yeah.
So that's the sad thing about some of the different, there's a break. What's that?
It comes out in this life.
Yeah, maybe, but it's not the same as, say, when people walked along and shit and kept eating and shit. And it's going to one big treatment plant is being treated. We don't even know it could be sterilising spores or killing off big soil fungi or...
It's all biological and natural anyway. And then it goes back on the land rather than in the sea,
Yeah,
which is
maybe.
where it usually would go.
I don't know. I grew up with an outdoor dunny. I'm quite
Yeah,
comfortable
me
with
too.
it.
It's the best way to
Yeah,
do
but
it.
it's a problem when you have a
Use it
million
on your-
people doing
I
that.
bought them online.
But if you have it, like everyone does the right thing, you never have a problem because that's how
The
millions
water,
of
the
years
cup.
of
Yeah, do you want to
evolution
take some?
happen. People should-
Because I've got
There's
another
never
bottle
that
at
million
home.
people
No,
in this one.
I'd
That's
get.
the problem.
I won't use it for this. I've been looking for it for
Whoa,
other,
that's what
other
it's meant to
jobs.
be.
Oh, you can have some.
But no,
Probably sterile within a second
it
there.
was mainly I
Need to
can
go that
buy it.
I don't know, it's supposed to go like the way I learned it, you're supposed to wait till
It was,
goes,
yeah, just online. I just put in colour changing silica beads.
Oh, is that all?
Yeah.
Now, is there another?
Lots of other silica beads come up, but this one was the only one that said it was colour changing.
Ah, that's the
It goes
key.
from white to
Right,
blue.
thanks.
I think it goes blue to pink.
Blue to pink.
Oh, right.
Yeah, blue to pink. When you know that it is. Yeah. Oh, thanks for that.
It's probably a 50 now.
Colour changing silica beads.
Oh, I like these tweezers. They've got
Don't talk over the top of it.
Martina. Oh, what's the temperature?
Well, it doesn't show.
Oh, sugar.
Have we gone overboard? With 10.
Must be close. I reckon it'll be just below. Let's see. It's right on 50. It's right on 50. Oh, good.
This is like food.
Okay,
This is
so
cool.
I just
saw
squirt
that
that
from a
straight in.
distance.
Stir it with my thermometer because it's already been in there and it's clean. And then before it cools, you start pouring as quickly as you can.
So it has to be done exactly at 50 degrees.
Yep, according to them. That's not going to work, Gil.
That's not.
They're all going to be crooked. If someone wanted to come and pop lids on, I would appreciate that.
I'm on pretty much straight away.
Yes, please. I was doing them a lot thicker, but Roger suggested to me when we were here last month that you really only need a very small layer in the bottom. It doesn't have to be much at all.
This is crazy. Staying a little sticky shit.
It's the fun chair.
I think I can get it off.
You guys are welcome to take some of these home to have a muck around at home, trying to get your own
This is so hard.
funguses going, and you
You're
can
doing a
always put them in
second
the
one
fridge
with
if you
the
get
same-
something
I want to do
going
the
and
spores, yeah.
bring it back next month and we could all post it together or something like that.
Ah, that's the red body.
I've printed plenty of bits of paper for you
The
to
other
take
way
it
we
home
could
and
just
get me
dissolve
write
the
down your own
spores.
information. Stop talking to me, you idiot.
Just spray this with alcohol. It's gas. Won't work. I'll probably go and wash them actually. Oh yeah, you're pouring a little. That's cute.
They're not that easy to
They're not
actually,
easy to
you know.
close.
But once they're
They
closed, they're
snap,
quite,
yeah. They snap on alright,
yeah,
but it takes
that little extra click, doesn't it?
a bit of practice. Yeah, well, I'm trying to keep the. Don't touch the inner side. Yeah,
No.
touch. Trying to keep the inner side protected before.
I mean, you could do this in like a box, right, as well, if you really wanted to be pedantic.
You could be so pedantic, but they reckon that hydrogen peroxide, Sandra, makes
got
this
us
something
so-
that you can
that's
make
so
up in the
awesome.
field.
That is just awesome.
I think it's both the
Makes
temperature because
sense,
it's still
hey?
50 degrees. It's still not.
That's right. And then the big hit of that H2O2
Spores that go in,
is just right, I think.
a little bit of COVID and
So there's plenty here for people to take home, muck around with. There's plenty for you to have, Sandra.
Cool. Patrick's coming next month,
Oh, really?
and he's coming to Bush Time, doing this. So he'll practice on us, and then he'll do it again with people that are not.
Can't wait to see him.
Yeah.
Oh, yeah. I've gone and got the permits to go into the state forest to get mushrooms now. So if anyone wanted to have a look at what that looks like as well. You get them through the Queensland Mycological Society. You've got to join the Queensland Mycological Society.
To get the what, sorry?
To permits.
Oh,
to,
permits, yes. That's very important.
So I
because
can
we don't
just
want
go
to
in
just
there
go
now and just get
pillage
little
the
DNA
jungle.
samples legally. Yeah.
You need to stick them in here.
into a state forest.
No, you need a permit
Well-
to collect them legally.
Oh, okay.
As in
Yeah.
national parks, not on
Yeah.
private land.
Stuff
For
all
private
the
land, you
time.
need an owner's permission.
Yeah.
So like we can get permission from the council because they own this land
But
now.
what about state forest?
Yeah.
Is state forest a national park or is it
I don't know if state forest. I
The-
guess so. Yeah. 'cause
The Queensland Mycological Society has a list that they've gained approval for.
Yeah.
Thanks, guys.
so these go in there?
Yeah,
That's
why
it.
not?
We've got everything.
I love the egg carton.
Let's try and close them even better because some of them I feel
Yeah,
like
no,
we
I
didn't.
noticed some
Just give them another press as we go.
Well, you just lift it at the top and you realise straight away whether they're on
Yeah.
or not.
That's true. That's very true.
So are these the ones we just poured here?
These ones.
These ones.
All of these, yeah, and these guys.
We'll just
I'll
put
pass
one
them
there.
over.
Can you put them in the fridge or?
You can, they recommend with all Petri dishes to keep them in the fridge. Funny thing is, I was doing that, and then because I
At least we know
was
where they are.
trying to find mushrooms, I had them in my backpack. They were there like 10 days, and then nothing happened to them, so I've just been leaving them out. They seem very sterile.
Yes.
Yeah. But I mean,
Actually, no.
if you thought you're going to need them 3 months, they might be sitting there, put them in the fridge, and then you don't have to stress, do
It's
you?
just really hard because when you move them, that shakes.
No,
No.
The liquid
no,
shakes.
no, well, same as those ones I've brought down. It's quite warm today.
Yeah.
Yeah.
It'd be fun to do this in a national park sometime with you all. So.
Yeah, that's what, like a proper foray, where you went out, wrote down where you were.
Yeah, for a weekend.
Yeah.
Lots of samples together.
Did you know the QMS does that already?
Yeah, but
if
where?
you join the
Not here.
Queensland Mycological Society.
Yeah, but nowhere near us.
Oh,
But
okay.
they will make one.
Yeah, well, we
Say,
need to be
I
real
want a
nice to
foray up
Patrick.
here and
Tina.
So what numbers should I put for the Aceroa rubra?
No,
Were we up to 7? No, 8.
So I'll
Six.
put them as like whatever it is, point something.
7, and then you sit here and fill out
Okay,
your
I'll
bit.
do that. Yep.
So there's
So 7,
Mycos.
did you say?
Yeah, hashtag 7.
I might stop doing this now.
Okay,
I
let's
still
stop.
found joining the QMS.
So are you calling those Petri dishes rather than Petri tubs?
I don't care what people call them. They have agar in them.
Just call it agar because people
I
get so
literally
confused.
went to the packaging shop and said I would like a small container, and this is what they sold me, which turned out to be perfect. So they're a plastic disposable dish I can post to Roger, I'm hoping. Yeah. I don't know. Yeah, this Petri dish. Petri. Is it maybe because I'm saying Petri dish is really a glass dish,
What's
isn't
the
it?
guy's name that created the dish?
Yeah. Okay, so what will I call these? Tina dishes.
No, just call it an agar.
Call it Tina dishes.
What?
An agar plate.
Yes.
It's an agar
Yeah,
plate.
that's exactly right.
Yeah, that's an agar plate.
A
Yeah.
plate or a pot.
It's a
Yeah.
Yeah, but it's the actual
Agar Tina.
I'm thinking you should pick one and then kind of it's your name.
Does it?
yeah. Okay.
Call it
Tina's
agar.
agar.
Call it agar.
Tina's agar.
I just call it
Tina's
a pot.
tubs. Tina's
the
tubs.
The dish is irrelevant.
So what are we doing, teens? We're doing
Spore.
yep. And what species was it from?
The gymnopilus.
The Gymnopilus. And we're doing a spore syringe.
Yeah, I haven't actually practiced this, but this is what they were telling us. So I've got some distilled water here that I made myself.
Yep. Here's what I prepared earlier.
Yeah.
Agar in there, but the agar is actually
And we'll just.
grown.
Okay, look at that.
Yep. There it is.
Okay, so then we're just going to suck.
Sorry, your agar.
Yeah, right here.
Oh shit, we lost our spores.
And in theory, this should only need 8 drops.
Billions of them.
Yep, and there we go. This is how people also store their spores. They put them inside syringes and leave them in their fridge. They can last for a year, sometimes 2 years. The spores will stay alive. So that has the spores in it, as you can see. And we've only added 1 very tiny drop to that. It's just distilled water.
Ah, yeah.
And so you did the spot thing on the output.
Yeah, so that you can
It's actually
then...
better, eh? They were telling
Yeah.
us in the course.
Yeah, so that's what they suggest to do, is to
The
do it on.
colour also is visible much more. Lovely earring.
And I shot my distilled water, and that's another really simple way to gather mushroom DNA, I guess, because that's going to hopefully start a very small gymnopiles in there. So yeah, I think that's it.
Yeah.
There's a lovely book.
You'll wait until that's grown before you send it off.
Yeah, that you know, it only takes. I only made these after the rain early this week. Sometimes it only takes like a day, and you'll look in there.
But from the spores it probably is a bit slower, isn't it?
I don't know. I'm sure if you googled it, there would be something in a mushroom growing site that would tell you. That, like I said, that's my first time of actually having a go at it, even though I'd seen it. Yeah. Woo. Thanks, Gilly, for bringing that. Okay, so yeah, everything's here to go forth and get some samples of whatever you would like.
Has everyone got a sharp knife?
I have 2. Oh, well, you don't need it till you come back. You know, they could just go find a mushroom, bring it
Yeah,
back.
I just really don't want to take mushrooms out if there's just one. Like, I really don't want to do that on the side. I want to continue that.
This is why everyone should have
lineage.
bought their own mushroom.
Yeah. Probably.
Unfortunately, I did mean to stop and get some oyster mushrooms. Yeah,
Yeah.
just do it out there. Take
Take a
these
small
with
sample,
you.
yeah.
There's a stick just
Because we could literally even cut it with scissors, you know, like a little patch of it.
Yeah, just bring that over, and we can take a little piece.
So then we can take the stick back again.
Oh yeah, if they're on
Yeah.
sticks, that's nice, but if they're in the ground, that's probably a really good
I
point.
have
Very
printed
good
that
point.
agar recipe for anyone that would like to take it home. Very simple.
Are you getting any bacterial contamination in them?
No,
Hydrogen peroxide, we've just done a whole workshop on how to do it sterilely. Yeah.
Okay.
it's been fantastic. Like I was saying, I was keeping them in the fridge, and then because I've been having them in my backpack, hoping for fungus, I'd forgotten about them. It was like 10 days, and I was like pulled them out, and they're just perfect still. So I haven't even been having them in the fridge.
Cool.
Been very sterile.
I think you're better off not putting it in the fridge, because if something grows, it will grow before you put your sample on.
Well,
If
fridge,
it's not sterile.
but the whole point of it is that the fridge temperature will reduce growth.
Yeah, but that's the point.
Yeah.
But if they're
Because you don't want to
contaminated, you
reduce
want to
the
know.
growth.
Ah, you wanna
actually-
see
if
it.
Something is contaminating
Right.
these
Yep.
dishes.
Yeah, you want to
You
see
don't want
it.
it growing.
Yep.
Yeah.
before
Very
you
good
put your
point.
sample on,
Oh.
because if it's growing,
Yeah.
it tells it
You
away.
definitely
True.
know when it's contaminated. Like anytime we do stuff together, laughing, talking, just be blue.
But if it's in the fridge, it's going to take a long time.
Yeah, so
for it
then
to
you
grow.
put it on and it's like something else has got a head start. No,
And
very
it's
good
taking
point.
off when the other one is on, and that's the
Yeah,
wrong
very
time.
good point. Yeah.
Because if it's sterile, it's sterile.
Cool. So should we
And I also
I'll go and get
printed
a stick.
one of these.
get a
Anyone
stick?
want one?
Yeah.
That's the
And then there's
information Roger would like.
The bracket fungi is still down in
Ooh, you're a good spotter.
the ones we saw
All right, let's do all of
months
them.
ago.
Yeah, that's good.
So I've got like agar powder.
All right, I'll stop the video now.
In that's sterile. I've had it in a pressure cooker, and you just leave, and then you get that when it's growing and it's got the culture in it. You can add it to your sterile grain, and this is perfectly happy mycelium that I have created from doing this process. From one little tiny piece of oyster mushroom to adding it to my sterile grain that I've sterilized in my pressure cooker. It has totally consumed that. If I was to let that air, it would grow a king oyster mushroom. And this one would grow a Queensland white oyster.
Cool.
These are, yeah, basically this one with the grain, 100 grams of grain will go with 1 kilo of pasteurized straw, and that's basically your little mushroom kit, edible mushroom kit that you pay for if you buy a mushroom kit. So that's
How do you pasteurize your straw?
with hydrated lime. So you just soak it for 4 to 6 hours. It's like a big tub of water with like half a cup of lime. Let it soak, you tip it out, and then you just add your
Make sure you
mycelium.
drain it so that there's no water dripping
Yeah, it's
off
got to
it.
be a pretty nice. Yeah, you let it sit for like half an hour, really, don't
Yeah,
you?
so it's just not dripping at all when you squeeze it.
Yep. And then 100 grams of whatever mycelium you've created, and away you go. And it works because I've actually done it.
So tell me about this more, because I missed the first part. So you basically created liquid spore culture out of it. Is that correct?
Yeah, so I have the liquid culture recipe there too. You have the marbles in it so that when you do inject, and you just need like literally inside a syringe, the tiniest bit from the needle, that you would push through your little hole, and it is silicone. And so when you pull your needle out, it will seal back behind you, which keeps it contaminant free. And you'll see it start to grow, and the marbles are just a cheap man's version of apparently a magnetic stirrer.
Yeah, fantastic.
So
Couldn't
that's
need all
it,
that
and
science
we'll,
equipment.
So what type of silicone?
it's like
A food silicone
exhaust.
or just
No, it's just exhaust silicone or engine silicone.
So it can tolerate heat.
Yep, so it can tolerate heat. So because when you put that whole thing in the pressure cooker,
Yeah.
and massively sterilize it.
And it's
Yeah.
stirring because of the glass beads being like boiled in the. Is that what you mean? Is that
I
why
put
it's crawling?
these were in there. I put the
Okay.
liquid culture in. I put the lid on.
Yeah.
I put alfoil over the top so that there's no moisture will get in. Pressure cook it for, I've written it down in my book, I don't want to say in case I get it wrong. And then you pull it out, I pull the alfoil off, I pop micropore tape straight away on my little breathing hole. And here I am waiting to find an edible oyster mushroom or any other edible mushroom. Have a great book here, everyone's welcome to have a look at. It has some of our local species that are edible. And it's a really good, it's the first Australian book that has edible mushrooms. So it's a very, very good book. Okay, that must be the end now.
All right. Until you come up with some other treasure.
Where do you get that micropore tape you're telling me about?
It's in your first aid kit.
Yeah.
Micropore tape is you put it on wounds and it'll still bleed.
Exhibit A, B, C, D,
Yeah, I've got some right here.
Oh, you've got the orange one.
Wow.
And a yellow.
Oh, yeah.
Perfect.
Yeah, yeah. So this is, yeah, it's really good for the mushroom, especially if you're making your own, growing your own mushroom at home.
Yeah.
Where you'd put your oxygen hole, put your micropore tape over it so it's still getting oxygen in and
Yeah.
stopping contaminants, and the mushroom will just push it out the way when it's ready to fruit. Yeah. Look at her socks.
Are there mushroom socks? Wow. I've got to take a video of that. Video socks.
And my shirt, actually.
Oh, that's so cool. Yay, mushrooms.
Yeah, and this is the information Roger would like.
Yeah, thanks for that.
Here, Doc.
Yeah.
And there's the
that's great.
agar.
Thank you very much.
Which, yeah.
And did you have your liquid broth?
It's, where's my book? Right here.
Is it this?
Oh, that was to tell myself not to talk too much while I was pouring the agar and stuff. Here.
I'm just going to